I'm going to talk about our efforts to generate antibodies with diagnostic and therapeutic potential for patients that have transthyretin amyloid. It's a disease associated with aging, and it involves aggregation of the transthyretin protein onto the heart or nerves of patients. This project was initiated by Stan Primer at the Supercentenarian Research Foundation because recently it's been reported that 7 out of 11 supercentenarians had significant deposits of transthyretin in their heart, and this may have contributed to their death. This project has two arms in a sense. My lab is focused on generating antibodies with diagnostic potential. Sudhir Paul's lab at the University of Houston Medical Center, he is focused on generating antibodies with therapeutic potential. So transthyretin is a protein that is found in the blood and the CSF. It has several physiological functions, including transporting thyroxine and retinol binding protein. The transthyretin molecule consists of 4 monomers together as a tetramer. Each monomer contains beta sandwich structure and beta helical component. And in essence, this is a molecule that consists of a dimer of dimers. The interface of the two dimers is where thyroxine and retinol binding protein bind. So in transthyretin or TTR amyloid disease, what happens is the protein aggregates, it deposits as beta sheet rich, ordered hair-like structures under electron microscopy, deposits with accessory molecules, and this process in itself, as you can see in a dramatic, I suppose, picture here of a heart, the sheer deposition and number of these deposits can affect the organ and is toxic to the patient. However, more recent studies also indicate that soluble aggregates of TTR may also be pathogenic. TTR transthyretin is associated with 3 different amyloid diseases that affect the patient's heart or the nerves. With the wild-type protein, it's associated with senile systemic amyloidosis. This is a disease associated with aging, where the patients are primarily those greater than 70 years old, primarily men, and it primarily affects the heart. Recent studies suggest that even as much as 20% of individuals over the age of 80 might have deposits of TTR in their heart. The disease is also associated with carpal tunnel syndrome, where early deposits of TTR can also happen in the median nerve in the wrist of these patients. A more aggressive form of TTR amyloid occurs with the mutated protein. This can occur as early as patients in their 30s or even younger. It can either primarily affect the heart or the nerves, and it's associated with familial amyloidotic polyneuropathy and familial amyloid cardiopathy. Currently, there's no cure for these amyloid diseases, and they are underdiagnosed. Current diagnostics for TTR amyloid are far from ideal. That's why we're trying to generate diagnostic antibodies. The main method is invasive and involves tissue biopsy. First of all, the amyloid deposits are stained with an amyloid reactive dye, Congo red, and immunohistochemistry then has to be carried out to make sure TTR caused the amyloid, because there's approximately 30 unrelated proteins and peptides in the body that can form different amyloid diseases. Less invasive diagnostics are under investigation. These include imaging regimes using proteins or small molecules that target amyloid, and also echocardiography, where deposition of amyloid in the heart gives a particular type of patterning. However, these diagnostic techniques are far from ideal, and they're not specific for TTR amyloid. That's why we're trying to generate diagnostic antibodies. Potentially, they could possibly be used as an imaging agent to image TTR amyloid, or to be used in a blood test for patients to detect aggregated TTR. Proof of principle for using anti-amyloid antibodies occurred recently by previous colleagues of mine at the University of Tennessee. They used a radio-labeled antibody 11-1F4 that recognized not TTR amyloid, but antibody formed by antibody light chains. This antibody recognizes a unique binding surface at the end of the aggregated light chain that's stabilized by a proline residue. They were able to show in a small clinical trial that this antibody was able to effectively image at least half of these patients where it deposited in different tissues and organs in the body. This image here is from a patient using CT PET imaging and shows that the antibody was able to detect amyloid in the lymph nodes. So although there's no cure for TTR amyloid currently, a lot is known about how it aggregates. And this brings up potential therapeutic targets. Basically, what's believed to occur is the tetrameric protein dissociates to form a monomer. The slightly misfolded monomer then goes on to form a substrate for aggregation. To date, there's been three main therapeutic approaches. One is to decrease production of TTR by gene therapy or liver transplantation, because patients that have mutant TTR, if you replace, in very few patients, I suppose, that are suitable for this, if you replace their liver with a liver that actually produces the wild-type protein, this can slow down the disease to some extent. However, the wild-type protein can still deposit on existing deposits. Also, we don't know the long-term effect of reducing the amount of TTR in these patients. Another approach is to increase the stability of the TTR molecule using small molecules. This has had limited but modest success so far. Most recently, a drug, tafamidis, has been approved in Europe for stage 1 for patients with mutant TTR. However, it didn't reach its primary endpoints, but it's able to reduce neurological deterioration in these patients by about 50%. And a third approach is to remove existing amyloid in these patients. And this is the approach we're focusing on. Sudhir Paul's lab is using a unique approach where he's using catalytic antibodies that act like enzymes that are able to digest up specifically TTR aggregates. This type of approach may have some advantages over classical immunotherapy, that this approach doesn't stimulate the cellular immune response, so you're less likely to get side effects such as inflammation. So Sudhir Paul and others a number of years ago actually discovered this particular property of antibodies, catalytic antibodies, that can be directed towards aggregates of amyloid proteins. Initially, he focused on Alzheimer's disease, and now we're trying to determine whether any of these antibodies have therapeutic potential for TTR amyloid. It is hoped this is an innate activity of antibodies that we all have in the blood. But Sudhir and others have shown that antibody to antibody, the catalytic activity isn't the same. It can be magnitude of orders different. So it is hoped to identify a particular monoclonal antibody that will boost the immune system of these patients and help clear TTR amyloid. So for the remainder of the talk, I'm going to focus on my lab's efforts to generate diagnostic antibodies with diagnostic potential that recognize aggregated TTR without recognizing the physiologically relevant unaggregated protein. And then I'll describe a few slides in Sudhir Paul's work on the therapeutic potential of catalytic antibodies. So the first step to generate antibodies with diagnostic potential was to stimulate the B cells in mice to produce antibodies against TTR. And to do this, we used two different immunogens, two aggregated forms of TTR, TTR fibrils, and also soluble aggregates of TTR, because as I described previously, it may not only be the amyloid deposits themselves, but soluble aggregates that may be pathogenic. We demonstrated by size exclusion chromatography that these aggregates were high molecular weight, and also they retained some of the properties of amyloid fibrils in that they were able to bind to an amyloid fibril reactive dye. When we immunized mice, we got a very robust immune response with these immunogens. In each case, antibodies were produced that were cross-reactive with TTR aggregates, whether they were fibrils or soluble aggregates, by ELISA. And even when we diluted out the serum of these mice 100,000 times, the antibodies still had very strong reactivity against aggregated TTR on the plate. We went ahead and carried out a series of screens, and we identified 9 lead monoclonal antibodies. We carried out a fusion, to generate stable cell lines, each of them producing a stable monoclonal antibody against TTR. At the end of this, we identified 9 monoclonal antibodies as leads, and this is just binding to TTR fibrils on the plate. And you can see here that they had binding activity from the low nanomolar to the picomolar range. In black are binding curves for some standard commercial TTR antibodies, showing our antibodies were just as good or slightly better, some of them, in terms of their reactivity. These antibodies also cross-reacted with soluble aggregates of TTR, and we also demonstrated that they reacted with in total they reacted with at least 4 different binding sites on aggregated TTR. So from now on, I'm just going to describe results from one antibody from each of the different 4 groups. Why we were characterizing these antibodies, to ensure that these antibodies did not recognize native TTR, but only recognized the aggregated protein, we had to come up with a barrage of different assays. Because standard ELISA, where, for example, you put native TTR down on the plate, can induce artifacts, antibody artifacts. For example, the protein can be partially denatured, or each antibody might bind to 2 different TTR molecules on the plate, enhancing binding. So we came up with a barrage of different assays, including solution phase ELISA. It was quite clear early on that we had generated antibodies that didn't recognize native TTR, but recognized TTR either when it was aggregated or when it had changed structure, when it was stuck on a surface. So in initial characterization, by just putting native TTR or aggregated TTR on the ELISA plate, our antibodies didn't distinguish, though, too well between different forms of TTR. This is our control anti-TTR antibody that looked quite similar. However, when we attached TTR to a different type of surface, nitrocellulose, we got a big difference, where our antibodies recognized much stronger aggregated TTR, while they had much less activity against the native protein. This was in contrast to our control antibody that recognized everything very similar, in a similar fashion. Likewise, when we conjugated TTR in a different manner using covalent coupling, using primary amines in the protein, we determined that some of the antibodies that we made didn't recognize wild-type native TTR at all, but they recognized a mutant form of the protein that is prone to aggregation. In stark contrast, our control TTR commercial antibody recognized everything similarly, giving us some evidence that these antibodies recognize cryptic binding sites only found on non-native aggregated TTR. To confirm this, we carried out solution phase ELISA assays, where, for example, in a capture ELISA, we put our test antibodies on the surface of the plate. We determined how well they could bind native TTR or aggregated TTR from solution. And what we demonstrated, well, first of all, with the control antibody, that it recognized everything essentially the same. In stark contrast, our lead antibodies didn't recognize the native protein. It only recognized soluble and fibril aggregates. This is just representative binding curves from where some of the data came for one of the antibodies, for example, and it shows with different concentrations it had essentially no reactivity with native TTR. A similar type of solution assay, but a competition assay, doing it a different way, where in solution we had antibodies, then we had either native or aggregated TTR to see whether the antibodies could bind to it, and it prevented the antibodies from binding to TTR aggregates on the plate. Here we had more evidence again that our antibodies were very specific for aggregated TTR, didn't react with the native protein, which was in stark contrast to this commercial anti-TTR antibody. Again, a representative binding curve for one of these antibodies, demonstrating that even at very high concentrations of native TTR, it had no effect on the antibody binding to TTR aggregates on the plate. So now that we had determined our antibodies had some of the properties we'd like for a diagnostic antibody in terms of specificity for aggregated TTR, I go back to the mechanism I showed earlier. We wanted to see whether these antibodies were able to recognize TTR monomers, because that's one of the first steps that's believed to occur, dissociation of TTR to monomers to initiate aggregation of the protein. So we isolated TTR monomers using a mutant TTR using size exclusion chromatography, but we demonstrated that none of our antibodies had any affinity for TTR monomers. As you can see here, only the control antibody recognized native TTR monomers and fibrils. Our antibodies only recognized the aggregated protein. However, just to show, under some instances there is reactivity. For example, when you put TTR monomers on a surface, now our antibodies reacted very strongly with it, suggesting they bind to a cryptic epitope on the non-native protein. So having determined the specificity of our antibodies in terms of recombinant protein, recombinant TTR, we want to see, did they actually react with TTR amyloid from patients? And to do this, we first of all carried out ELISA studies, where we just put the amyloid on the plate and determined antibody binding curves. And what we determined was all our antibodies were able to bind to the amyloid. They varied in their ability from essentially about 100 nanomolar down to activity down in the 1 nanomolar, subnanomolar range. In stark contrast, a commercial antibody against TTR had no effect. That antibody was made against native TTR. To confirm that this activity was against TTR and the amyloid, we carried out competition studies with our antibodies. For example, with this antibody here, we're able to demonstrate that TTR fibrils and soluble aggregates of TTR were able to prevent the antibody from binding to amyloid on the plate. We then chose our best antibodies and determined and showed that they were able to bind to amyloid on tissue sections from patients that had amyloid in the heart. And this is one of our antibodies here, and it shows it strongly stained amyloid in the tissue, based also on the staining of an amyloid-reactive dye, Congo red. In contrast, the antibody had no reactivity with normal heart tissue. So in terms of the diagnostic antibodies, we've demonstrated that we've generated some lead monoclonal antibodies. They appear to have the properties we want. And I forgot to mention actually as well, these antibodies are active in human serum. They were still able to bind to patient amyloid in the presence of human serum, which has high concentrations of native unaggregated TTR. They react with aggregated TTR, and they didn't react with the native protein, which we want. However, we're really at the beginning of this, and we really have to determine the diagnostic potential of these antibodies in vivo using a mouse model of TTR amyloid. Also, another possibility for diagnostics is to determine whether any of these antibodies can be used as in essence, in a blood test. Can any of the antibodies detect TTR aggregates in the blood of patients? So just for the next few slides, I'm just going to describe a little bit of the work in Sudhir Paul's lab, where they've been focusing on identifying antibodies with therapeutic potential that can catalytically digest, just like an enzyme, aggregated TTR into harmless species. So the initial studies they carried out was to determine what class of antibodies was best for degrading aggregated TTR. So from their previous work and others, they have demonstrated that this is an innate property of antibodies. It may be involved in the defense mechanism against misfolded amyloidogenic proteins. So first of all, they determined from pooled antibodies for normal individuals whether IgMs, IgG, IgA had the best catalytic activity, and they demonstrated that IgMs were essentially the only ones that had very good activity. With the radio-labeled aggregated TTR protein, the IgMs were able to digest up the protein as seen here in SDS page down to much smaller fragments from the 7 to 13 kilodalton range. The IgMs, in stark contrast, these IgMs had no activity on native TTR. They were specific for the aggregated form. And as I mentioned, IgAs, IgGs had essentially no effect. They went ahead then and screened human monoclonal IgMs from patients that had cancer of B cells. They were able to get a supply, and they identified one lead candidate, an antibody 1802. And they demonstrated, like the pooled IgMs, that it was able to cleave up aggregated TTR, was specific, didn't react with the native protein, had activity in human plasma, and was specific. It didn't cross-react with any other targets. Just the examples I'm showing here demonstrate pre-aggregated TTR, that this antibody by turbidity assay was able to essentially reduce all the aggregates down to nothing, and a control IgM had no effect. Also encouragingly, they demonstrated that the products from this reaction doesn't seem to have amyloid-like properties, in that amyloid-reactive dye, thioflavin P, didn't have any reactivity with the reaction products from the antibody catalyzed reaction. So Sudhir Paul's lab is continuing to characterize the catalytic activity of these, the lead antibody. He also hopes to generate other antibody therapeutics using technology they've developed, where they can actually stimulate the immune system to make better catalytic antibodies, and also to isolate these antibodies from phage display libraries. And it is hoped that hopefully at the end of this that we'll have gained information to facilitate the development of new antibody therapeutics to benefit patients with TTR amyloid. And I'd just like to thank all these people here and SENS Foundation for funding. Thanks very much.